students. In this video, we are going to learn about artificial hybridization. And in this technique, we are crossing two genetically different individuals to create a new genotype by controlled pollination. And artificial hybridization is the major approach of crop improvement programs. This process involves two steps. First step is emasculation. As this picture shows emasculation, and in this technique there is a removal of anther from the flower before they dehisce the pollen grains to the stigma of the same flower, that is called as emasculation. With the help of scissor or with the help of forceps, we are going to remove the anther before they mature to release the pollen grain. The second step is bagging. After the removal of anther, the flower stigma, particularly stigma, is going to be covered by butter paper or polythene paper to prevent the contamination of stigma from the unwanted pollen grains. This is called bagging. So the first step in the artificial hybridization is anther is removed in bud condition from the bisexual flower, that is called as emasculation. After emasculation, we will get a female flower, and that female flower, that emasculated flower, is enclosed in a bag made up of butter paper and plastic. The technique is called as bagging. After the removal of anther, the stigma or the flower has to be covered by polythene bag or butter paper. Then mature weighable pollen grains are collected from the male plant. From the another male plant, we have to collect the mature and weighable pollen grains, and bag is to open and pollen grains are dusted on the stigma. We have to remove the bag and we have to dust the pollen grains on the stigma. Then immediately we have to replace the bag, and then we will get a artificial pollination is taken place.